Good morning. Welcome to the Harmful Algal Blooms webinar series. My name is Amy Weckel, and I'm from the Illinois Water Resources Center at the University of Illinois in Urbana-Champaign. I will be your moderator today. This webinar is hosted by the Algal Bloom Action Team. Let me see if I can get my slide to go. Here we go. This webinar is hosted by the Algal Bloom Action Team, of which the Illinois Water Resources Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to hosting a webinar series, our team is developing a website of resources, including fact sheets and frequently asked questions on HABs topics. I encourage each of you to visit our website, which is listed here on the slide. And there you can explore some of the resources we have to date, including a What You Should Know fact sheet, a HABs frequently asked questions database, and recordings from previous webinars and our virtual HABs research symposium, which was held in January. Before we get started, just a few housekeeping items. We will have a Q&A session after our speakers present today. Please list your questions for our speakers in the Q&A panel, and we will do our best to address all of them following the presentations. If there is a question already posted to the Q&A and you have the same question, please upvote the question and we'll be sure to get answers to the most popular questions during the session. Today's presentations are being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you're having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well, and we will be happy to assist you as best we can. Today's presentations focus on predicting harmful algal blooms, and we have two great speakers lined up for you. Our first speaker is Justin Chaffin, a senior researcher and research coordinator at Ohio State University and Stone Laboratory. Justin is the research coordinator for Stone Laboratory and conducts his own research on cyanobacterial blooms in Lake Erie. Justin's duties as research coordinator include arranging visiting researchers' visits to Stone Lab, coordination of Stone Lab's REU program, upkeep of Stone Lab's analytical equipment, maintenance of the lab's atmospheric mercury monitoring system, and serving as the lab's chemical and laboratory safety officer. Justin is also a licensed captain by the United States Coast Guard. Justin has lived year-round on South Bass Island and has worked at Stone Lab since 2012 after graduation from the University of Toledo with his PhD. Justin's presentation, entitled Microcystins in Lake Erie, Working Towards Developing a Toxin Concentration Forecast, will kick us off today. Justin, thanks for joining us. I will stop sharing now and you can take it away.
Okay, great. Can you hear me?
Yes.
Okay. And then my slides should be popping up now, correct?
We see them. We see.
There we go.
There you go. Yep.
Okay. All right. So, all right. My presentation is going to be focused on Lake Erie, but I do believe what we're learning in Lake Erie can be applied to any lake, large or small. Lake Erie, we have a good handle on forecasting bloom biomass, but not the toxins they produce, microcystins. And I'm going to be talking about the challenges we have at forecasting microcystins in Lake Erie. So a brief outline. Some of this might be a background or review. I'm going to talk about what are microcystins, a relationship between microcystins and bloom biomass, triggers of microcystin production. Can we make maps of microcystins that are kind of analogous to a satellite image? And then challenges to developing bloom forecasts. So microcystins are a group of toxins produced by cyanobacteria. Most of you know they target the liver. Microcystins need to be measured from water samples. We can't measure them from remote sensing. There are over 200 forms or congeners of microcystins, and their structure is overall pretty similar but their toxicity of each congener is very different. For example, microcystin LR is very toxic. Microcystin LR is the one that all the health guidelines are based on. But we also have microcystin RR, which is relatively low toxicity. There are several types of cyanobacteria that can produce microcystins. In Lake Erie, we're mainly focused on Microcystis, but we also have Planktothrix in nearshore zones and in some of the bays. We do have Delicospermum in Lake Erie, but it is not a microcystin producing strain. So all my information that I'm talking about today is dealing with Microcystis. A question that's commonly asked, is there a relationship between bloom biomass and microcystin concentration? This is data we collected for several years in Lake Erie. Cyanobacteria bloom biomass as chlorophyll A on the X, microcystin concentration from the ELISA method on the Y. And you really see there is no direct relationship between cyanobacteria biomass and microcystins. And if we zoom into where most of the data points lie, you really see there is no direct relationship. So if we're trying to develop a microcystin forecast, we cannot simply rely on bloom biomass. And as I mentioned a couple slides ago, that there's different congeners and those congeners have different toxicities. Microcystin concentration does not equal bloom toxicity. And I'm not going to get into that. I'm just going to focus on microcystin concentration for today, but we are working on toxicity. So why is there no relationship between bloom biomass and toxin concentrations? Well, within a community of Microcystis, there are toxic and non-toxic strains. Only genetic methods can tell the different strains apart. There are 10 genes that are needed to make microcystins. And if you lack one of those genes, it's rendered as a non-toxic strain. So you can have toxic and non-toxic strands within a population or even within a water sample. But also those toxin strains can alter the amount of microcystins they have per cell. So for example, this cell, cartoon cell here, has a lot of microcystins. This one has a little bit. This only has very little bit, and these two would be non-toxic strains. So we took the approach to understanding the relationship between the microcystin to biomass ratio. And if we look at a seasonal pattern and in Lake Erie, we see that the microcystin to biomass ratio decreases. That's what's shown in this plot here in the red triangles, showing microcystin to cyanobacteria chlorophyll A decreasing throughout the summer. And that pattern follows nitrate concentrations. We see that every year in Lake Erie. And if you look at the gene pool, we see that early bloom, when there's a lot of nitrate around, we have more toxic strands. Between 70 to 90% of the cells out there have the genes. And then we get a shift in the community to mostly non-toxic cells. And we also know that light has a role in this too. So you have clear water, lots of sunlight early summer, muddier water, less light in the fall. So the decreasing nitrate and decreasing light levels switch from toxic strands to non-toxic strands. And we also can do experiments where we spike in nitrogen and we see the bloom producing more microcystins. And we also know that high light levels are needed in these experiments. So at low light, we do not see the effect of nitrogen. We only see that effect at high light. So at a large scale, cyanobacteria biomass can be monitored with remote sensings, but we have, can we make similar maps for microcystins? These satellite images are really good, but they don't show you toxins. So can we make similar maps for microcystins? Well, as I mentioned earlier, we need water samples. So to make a high resolution toxin map, we did the survey, which we called a HABs grab, where we had 8 different boats out on the water from US and Canada, many different universities and agencies involved. And we collected samples on the same day within 6 hours of each other. All samples were analyzed by one lab. So we don't have to worry about different labs giving different results. So this was in 2019, August 9th, 2019. And then we did a smaller version of this HABs grab a year before in 2018, also early August. The bloom was much smaller. So the background for both of these slides are bloom biomass from remote sensing, and then the dots indicate where the samples were collected. So lower biomass in 2018 than 19. This was what I call grab and go sampling. There was no onboard filtering, no onboard processing. We just grabbed the water with a 0 to 2 meter integrated tube sampler and poured them into a 2-liter bottle. And then we used the YSI calibration cup to measure temperature, dissolved oxygen, pH, and those things. 2018, we collected 100 samples. In 2019, 172 samples, all in one day. It's just an example of a processing center. All the samples went back to a lab and they were processed. So here are the maps with the ELISA method for total microcystins. We see that just look in the 2 years, there's different range in concentrations and the pattern is different. So in 2018, we see higher concentrations along the entire length of the Ohio shoreline and up near the Bass Islands. In 2019, the highest concentrations are in Maumee Bay, up into the Michigan waters and along Ohio. And then we also had what we called this finger of toxins and finger of biomass that kind of swooped up towards Canada. Looking at 2019 in particular, we see that concentrations varied by 2 orders of magnitude over short distances, both at the bloom boundary and both in Maumee Bay, where we go from high concentrations to medium concentrations. And these rapid gradients are difficult for models to pick up. Talking about the microcystin to chlorophyll ratio again, we see that was not spatially consistent. So the cells in 2018, the bloom, in this area near Oak Harbor and out by the islands, these cells here had more toxins per cell as the rest of the basin. Whereas in 2019, we see higher toxins per biomass hugging the shoreline really tight and lower further off into the center of the basin. So if you're making a toxin forecast, you cannot simply rely on a consistent spatially or temporally microcystin to chlorophyll ratio. And we also see that ratio correlated with nitrogen again in 2019. At low nitrogen, lower toxins per cell, higher nitrogen, higher toxins per cell. We did not see a similar pattern with phosphorus or the NDP ratio or any of the physical parameters. So again, nitrogen is important for toxins. All right, so that was good. We made those high resolution maps with a lot of samples on one day, but can we use historical data to map microcystins from previous blooms? Fortunately, in Lake Erie, there's lots of data from water treatment plant operators who are collecting samples weekly, a lot of researchers collecting weekly to monthly, and then we partner with charter boat captains who collect samples weekly. So there's lots of data available in Lake Erie to make these maps. However, when we got into the data set, we really see that it's limited spatially. On this date in 2016, there's 16 samples collected. That's pretty good, but yet we still don't know what's happening across these large distances when we only look at microcystins. And it's even worse, it's a couple of days later, in August 4th, when there's only 2. So really, what's happening between these 2 points is an issue for models and for forecasting toxins. So what we did to try to overcome that is we compressed all the data into 1 week intervals. So example from August 14th, 2017, that'll be the week starting August 14th. A lot of toxins here, but when we look at bloom biomass from remote sensing, we see that they're not aligning. Like the spatial pattern of microcystin is not matching the spatial pattern of bloom biomass. And a couple weeks later, we see this example of that spatial pattern microcystin. It does not match that of bloom biomass. However, when we looked at the HABs grab data, we do see that the pattern of microcystins match the pattern of bloom biomass. So this tells us that routine monitoring of microcystin in the Western Basin, even though it is quite extensive, does not have the spatial resolution needed to inform models on a basin-wide scale. On the HABs grab, there was 172 samples, whereas this weekly data set, at the most 20 samples collected, and that's just not enough resolution. And where there's this difference between the 2 images is that there's just simply no samples collected here. So we can't extrapolate what's happening here to out there. So some challenges to forecasting these, the low amounts of data to initialize the models. And then once we run the models with bad input data, we're comparing that, I don't want to call it bad model, but not best as good as it should be. We're comparing it to not good data again. So that's an issue. The bloom boundary layer and rapid concentration gradients are difficult for the models to capture. There's no relationship between concentration and biomass, and the biomass toxin to biomass ratio is not spatially consistent. So we can't take a water sample that we know this ratio and extrapolate out to the unknown areas. So how do we build a forecast? For Lake Erie, we can forecast bloom biomass using phosphorus loading from the Maumee River. However, there's no relationship between river load and microcystins in the lake. So we can't issue a similar Lake Erie microcystin forecast yet. NOAA researchers recently did a hindcast exercise where they used the microcystin to biomass ratio that I said was, not the best thing to use. But then they just took, what's the probability of microcystins exceeding a concentration given a level of chlorophyll? What my group was trying to do was take these microcystin maps that we made, even though they're not entirely accurate or best that they could be, and add those into lake circulation patterns. So if we know where microcystins are today, and if we know where the water currents in Lake Erie are moving, we should be able to forecast microcystins several days out in advance. And then we can compare, what was modeled to what was observed. However, the lack of data, as I mentioned, hindered our start and our comparison output. So we then refined our effort to a smaller area. See, I'm running out of time, but almost done. We also built in microcystin production rates, where we measured production rates experimentally. And what I'll just cut to the chase in saying that models with microcystin production rates perform better than models without microcystin production rates. I'll skip through this. So suggestions that we need to improve forecasting capabilities. With the absence of microcystin data between 2 locations, using the microcystin to biomass ratio of nearby sampling points And then biomass data is likely the best current method to estimate microcystins at an unknown position. So at 2 points, you need to know microcystin and chlorophyll, and then you extrapolate that ratio in between. If they're nearby, not far by, really far apart. And then currently forecasting the probability of microcystins exceeding, or more simply, will microcystin increase or decrease over a certain time period? And especially we need more data. And we need to know more about production rates and the role of light. So with that, I'll stop. Thank the NOAA EcoHab program and NSF and NIH for funding this project. And I'll take questions when at the end. Thank you.
Thank you, Justin. That was great. All right, let me share my screen again. Okay, our second speaker today, there we go, is Katie Foreman. Katie is an environmental protection specialist with the US EPA's Office of Groundwater and Drinking Water and leads the office's work on harmful algal bloom and cyanotoxin issues in drinking water. Katie also serves as a source water quality technical expert in the evaluation of unregulated contaminants and national primary drinking water regulations. Prior to joining the EPA in 2015, Katie led water infrastructure projects with the Oregon Department of Environmental Quality, served for 5 years as a scientific and technical expert on Chesapeake Bay watershed water quality issues with the EPA's Chesapeake Bay Program Office, and worked for 6 years with the Iowa Department of Natural Resources Watershed Monitoring and Assessment program focusing on HABs, nutrient, and microbial non-point source pollution. Her presentation, entitled Effects of Harmful Algal Blooms on Regulated Disinfection Byproducts, Findings from 5 Utility Case Studies, will be our final presentation for today. Don't forget to put your questions in the Q&A panel and we'll address them following the presentation. Katie, the floor is yours.
Thank you, Amy. Okay, I'm going to share my screen now, and hopefully you all can see this. Everyone seeing my screen?
Yes, it looks great.
Great, thank you. And thank you for the opportunity to talk with everyone today and present on this work we've done here at EPA. And I really want to thank Justin for his presentation. It lines well. We're different topic areas, but very much related. I'm going to be talking and kind of shifting the focus a little bit more from the ambient water quality monitoring and predictive modeling to impacts on drinking water utilities. As mentioned in my intro, I work with the Office of Groundwater and Drinking Water. I am the harmful algal bloom and cyanotoxins lead for the agency on drinking water issues. So very interested in topics related to how can one be ready for harmful algal blooms in their drinking water systems. But one of the things I'm going to talk about here is also some of the unintended consequences of harmful algal blooms that may impact drinking water as it relates to when you have a harmful algal bloom in your source water and how utilities, drinking water utilities specifically, are managing multiple treatment objectives. So just going to mention this disclaimer. This is obviously, this presentation is mine alone, doesn't represent the policies of EPA, but want to just note a little bit on this conceptual diagram about this study objective. So this was a concrete study that we did to really explore these ideas of the impacts of source water quality during harmful algal bloom events, and how does that link to treatment outcomes and drinking water, your finished water quality. And a couple notes, as I know that many people probably are harmful algal bloom experts here in this group, and we probably have some drinking water folks as well. But for those of you that don't work in the drinking water world and day in and day out, Disinfection byproducts are harmful products that are produced and can be produced as a byproduct of the disinfection process, which, as everyone knows, is very important in our drinking water treatment. However, we do have regulatory rules on determining and keeping systems in check in terms of the levels of disinfection byproducts that they actually develop as part of the treatment process, and we're actually in the process of potentially revising those rules. So just wanted to note the context that we're working within here is looking at trying to manage two health outcomes based on treatment of drinking water, and the health outcomes obviously during harmful algal blooms are cyanotoxins and their direct human health impact if at elevated levels and consumed in drinking water, and disinfection byproducts that also have a health impact as well. And so we really wanted to get a sense of what we heard in the literature, and this is, I'm not going to go through this conceptual diagram, but the literature is really clear that in a lab setting, you have a harmful algal bloom, you have total microcystins, you hit that with chlorine in a lab, it develops disinfection byproducts. It's well established in the literature, and we call that that HABs are a precursor to disinfection byproducts, and that's well established in literature. But the degree to which treatment at the full scale can impact that lab experiment is what we wanted to look at here. And there are not many studies that do that. So we took a stab at this back in 2018, and we just published these results, and I'm excited to share them with you. So a little bit so you can understand some of the terminology. As I've mentioned, precursors related to disinfection byproducts. Those can include what's regulated precursor, which is total organic carbon. Also can include, as I mentioned, harmful algal blooms, total organic nitrogen, and other kinds of information. And source water conditions can impact the treatment and treatment outcomes. But there is a lot that a drinking water treatment system can do in terms of decreasing disinfection byproducts and cyanotoxins in their system. And then, that can lead to variations in finished water quality. So the study approach was looking at 5 utilities that were wonderful to work with and volunteered their time and their effort to collect samples for us. And these all had histories of harmful algal blooms in their source waters. And 3 utilities were in Ohio, 1 was in California, and 1 was in New York. And they all served a relatively small amount of people. Some were smaller systems, and the largest system was 55,000 people served. And we collected samples, as I mentioned, during the harmful algal bloom season in 2018, which ranges through December these days. And we collected samples once or twice a week. There are issues with collecting them in more frequency, so we did our best to collect as many as we could. And then we collected, important here is they're paired samples, they're raw water intake samples, so at the tank intake to the treatment plant, and then that represents the source water quality. And then we had samples for the entry point to the distribution system, so that's where the pipes go out to serve customers. And what we did there was we did a simulated distribution system sample, because the water that comes out of the entry point to the distribution system goes through lots of pipes before it hits the consumers. So we did a simulated test to try to be sure we accounted for the water quality that might result with that kind of time and exposure to oxygen, those kinds of things. So just to note here, for those of you who aren't treatment experts, I'm not going to go over this in a lot of detail, but those of you who are treatment and drinking water folks, you'll quickly see that these utilities have differences in their ability to treat these competing factors, harmful algal blooms, cyanotoxins, and disinfection byproducts, but there are a lot of similarities. So they have powder activated carbon available, and they also have ozone, and some of them have granulated activated carbon. This is important notes because I'm mentioning these are effective removal approaches for cyanotoxins and for disinfection byproducts, and they all have free chlorine as their disinfection. I'm not going to go over this in a lot of detail, but just to mention there are some differences, but there are some similarities as well. And as I mentioned before, we did raw water sampling, so source water sampling, and finished water sampling. And as I mentioned, those finished water, that information was held for 24 hours in a simulated distribution system situation to be able to mimic a distribution system for those results. And we measured chlorophyll A, total microcystins, and total organic carbon in the source water, as well as cyanobacteria cell counts. And then we focus on the two regulated disinfection byproducts, HA5s and TTHMs, and that was in finished water, along with looking at total microcystins and total organic carbon in finished water. So just really want to highlight the results. I'm going to go right to the results, and I can chat with other folks about our methodology as necessary. We did a lot of analysis on this, had some great data analysts on this project that did modeling and did multiple things to try to determine what we could find from the results. But in the end, some basic correlation analysis and data visualization was really helpful for us to understand these results. And here you'll see the results of this correlation analysis. We have source water information on the left, going down vertically, and then horizontally, we have source water, just total organic carbon. So that is, and I'm going through this quickly, but total organic carbon is the regulated precursor for disinfection byproducts. So it's a great indicator of whether or not we're seeing elevated precursor levels in these particular case studies. And we saw a relationship that was observed, and one would expect from the literature, that we saw at 4 out of the 5 systems. We have the source water total microcystins, cyanobacteria cell count, and chlorophyll A, one of those, each one of those systems being correlated with total organic carbon. So it really does confirm this idea that HABs are precursors to DBPs, which is not surprising. So the second result we found was that when you start to look at the relationship between the source water parameters and finished water, which is what the literature says, you should see a direct relationship between these. More of these should have more disinfection byproducts, because these elements on the left serve as precursors to developing disinfection byproducts. We are seeing that that isn't across the board. If the bench scale studies played out in the real world, we would see very similar to the results we saw on the last slide, where we are seeing these correlations, in most of these utilities. But as you can see, we're only seeing it in 2 utilities, and it's for that cyanobacteria cell count is what we're seeing a relationship with for the finished water disinfection byproducts. And there are lots of reasons for this, but I want to note in particular, total microcystins is a lot of work being done to relate that in source water to disinfection byproducts. And we're not seeing that here in this study for potentially multiple reasons. But one of the things that we really explored in this study was that, we aren't in a lab. We have treatment approaches in place that are trying and focused on these contaminants and trying to keep DBPs at bay and also manage cyanotoxins. So we did a little bit more digging into, just taking a look at, across the board, are these utilities, what kind of source water are they managing for their finished water? What kinds of additional variables are at play here. So I'm not going to go into this a lot of detail, but this is a normalized, this is a graphic to really show normalized distribution of different source water parameters as they relate to kind of action levels or levels that we really just use for benchmarks to consider high. And that's that horizontal red line. So anything above that, the distribution of those, you would consider that to generally be having high levels of that across the board. And we saw that these utilities had varying degrees of complications in their source water, if you will, to deal with in treating. But I really want to hyper focus you all on this cyanobacteria cell counts particular part of this graph, where you see all of them are above that level of concern in source waters. So this is to be expected. These utilities, have historic issues with harmful algal blooms. It's why we chose them. So it's not surprising. But what we dug into a little bit more is that we said, okay, so, these utilities all have these issues, when are we seeing what's the distribution of seeing elevated levels of cyanobacteria and toxins? And this kind of gets back to Justin's conversation. Obviously here, it was very hard to predict predict when microcystins are going to be a hit in source water. And you can see it's sporadic because, there's a lot of different factors that go into whether or not a bloom is toxic. But we thought it was interesting to ask utilities, when do you consider yourself having a harmful algal bloom in your source waters, thinking that might help them inform how they do their treatment. And what was really interesting is that there's no obviously consistent application of a definition for harmful algal blooms at these 5 utilities. But some of them said they never had a bloom event. For instance, C and D, you can see they have elevated levels of cyanobacteria across the entirety of the study. They have hits of microcystins, but no bloom reported. So we were just wondering, this doesn't necessarily mean they aren't effective at treating. This definition doesn't necessarily lead to other treatment approaches or decisions, but it could. So we looked at that and we were really interested in that. What I want to make sure folks understand from this study is that this wasn't a study showing, all the violations that these utilities had for whatever contaminants. All of them were really keeping none of them had total microcystins in their finished water, which is to be expected. They all are used to dealing with harmful algal blooms and have known effective approaches on their plants to actively treat them. But I also just want to note that you'll see this horizontal line indicating elevated levels. For instance, these disinfection byproducts, those 2 at the end, TTHM and HA5. For the bulk of this study, these utilities, really kept those numbers down. They were co-managing this contaminant, but to varying degrees. As you can see, there's a lot of variability, and a lot of what it came down to were operational changes that utilities use to manage them. And obviously, you can't compare directly, but we were really interested in the approaches these utilities took in some of the specific treatment approaches, such as powder activated carbon dosing and chlorine dosing. So powder activated carbon dosing is a really effective approach for managing DBP precursors and also for managing cyanotoxins. So we were just really interested in how they manage that. And we're really lucky to get their operational data. And you can see there's a lot of variability in how they dose this PAC. Some of them, for instance, dosed really high PAC, just one level the entire time. And you can see the wide range of TOC removal that's on the y-axis and is a good measure of effectiveness of that treatment approach. And you can see others really tailored the treatment to the specific water quality they were seeing. They had more consistent TOC removal, and it was a very different approach. So that really played into things, and we thought that was very interesting when we started to take a look at all of the operational data. And what folks might want to know too is that what about the chlorine dosing? So chlorine dosing, obviously, the more chlorine you put in, the more chances you have to develop disinfection byproducts if you aren't, managing that in other ways. And, often the way to manage a cyanotoxin bloom event is to hit it with as much chlorine as you possibly can. So part of the way to look at this is we were thinking, are we seeing this unintended consequence that chlorine is being used more during these harmful algal bloom events, and, what is happening to the disinfection byproducts? And, it's a very nuanced answer, obviously not as easy answer as one might expect. On the y-axis here is basically the yield of one kind of DBP, TTHM, standardized across all utilities. So you just think of that as that being more of the finished water disinfection byproduct that you don't want. And then you look on the x-axis, and that's a chlorine dose. The more chlorine, usually the more yield you have of disinfection byproduct. So you can see there are some systems that just hit it as hard as they could with chlorine, and they were managing and having more yield of disinfection byproducts, while others try to keep their chlorine low, which is a great way to manage your disinfection byproducts, and just adjust it a little bit, and had little variation and not a lot of spikes in their disinfection byproduct formation. So it really just varied widely. You can see the utilities that kind of set their controls and went, and you can see they have a lot of variation in the outcomes there. So just to wrap up I want to be sure there's time for questions just really quickly here. As I mentioned before, really 4 key findings. We did confirm with our case study that HABs in source water contribute to DBP precursors, especially and particularly TOC, and that source water quality harmful algal bloom parameters and finished water DBPs didn't consistently correlate, which is not consistent with the laboratory studies. And a lot of variation existed in how well they were managing both cyanotoxins and disinfection byproducts based on key operational adjustments, and potentially on whether or not they were, what they were seeing was happening in their source waters, potentially whether or not they had toxins or not. Those kinds of things could have also played a role. But as I mentioned before, these key operational adjustments, I think is really important to note that, the lab results show, increase this, you increase this, but there is that treatment option in the middle, and there's a lot of opportunity to co-manage these contaminants and do it really effectively. It just requires a lot of some pretty nuanced adjustments here and there, but we think we learned a lot from this study. And I just want to note there are limitations to the study, obviously not nationally representative at all. And I also want to note too, as the most important part of this, is that we didn't look at unregulated disinfection byproducts. And if any of you know the research, harmful algal blooms are in the research shown to increase these unregulated disinfection byproducts. So looking more into that, I think is great research to think going forward, and also looking into composition of some of the source water in more detail could be helpful as well. And I just want to acknowledge my co-authors and note that this just recently got published, this work in AWWA Water Science. So please look to that if you're interested in learning more. And my contact information. Thanks for your time.
Thank you so much, Katie. Let me go ahead and share my screen again. Okay. I'd like to thank everyone for participating in the Algal Bloom Action Team's webinar series. A special thanks to our presenters, Justin and Katie, for sharing their research with us. And also thank you to Ann Nardi and her team at the North Central Region Water Network for hosting the Zoom webinar and coordinating the technical aspects. We couldn't do it without her. As we move into the Q&A session of the webinar, I'd like to remind you to check out the Algal Bloom Action Team's website, and there you'll be able to connect with the team and subscribe to our events. While this webinar is the final event for the Algal Bloom Action Team in 2021, please join us at our second annual HABs Virtual Research Symposium, scheduled for January 6th and 7th, 2022. We have 16 cutting-edge HAB research presentations lined up, focusing on 4 topics: HABs monitoring and ecology, cyanotoxin treatment and detection, forecasting and modeling HABs, and emerging technology for detecting and monitoring HABs. You can register for this event now for free on our website. Finally, our webinar series will continue bi-monthly throughout 2022, beginning February 2nd. We will now move into the Q&A session of the webinar. We will draw initial questions and comments from those submitted in the chat box during the presentations. We will do our best to get to all of them. Following the Q&A, please complete the survey at the link being provided in the chat, and we appreciate any additional comments or feedback you have for this series. Okay, so let's see what kind of questions we have. Okay, if microcystin concentrations are impacted by high nitrate nitrogen, what happens when Lake Erie achieves reduced phosphorus and thus increasing the nitrogen phosphorus ratio? Is that a relative increase in nitrate nitrogen, which you imply more microcystin in the water column? Justin.
Yeah, that's a good question. And I know there's some groups working on that, and I know there's a paper in review at Science that I've seen early, or it's not published, but it's a modeling based exercise, and it does suggest that there could be, even though cells might be lower, you might have lower biomass if you reduce phosphorus. Those cells might actually have more microcystins per cell. And then does that equate to an overall increase in toxins? We're not sure yet. But we do know if you reduce phosphorus, you'll reduce the overall biomass. But the question about do the cells become more toxic or not, I think it's still under debate. However, I do think that a lot of land use practices that are aimed at reducing phosphorus might also reduce nitrogen by some by some ratio. I don't know if it's not going to be a 1-to-1 proportional increase, but it's going to be a grand experiment if we even do meet those phosphorus reduction goals.
Great, thank you. Do you also observe the effect of high nitrate nitrogen on the type of microcystin congeners produced?
We do, yes. There's two things that go into which congeners are produced. Well, one is the amount of nitrogen, and early summer when there's more nitrate around, we see more RR, which is about 17% nitrogen by mass. Late summer, when there's very little nitrogen available, we see more LR. We see more LA, which is only 10% nitrogen by mass. But also in that, there's a shift in the genome. Even within the MCYB gene, there's different populations, and then those different populations are producing different congeners. So yes, there is an impact on nitrogen on congener type.
Okay, and can we only use nutrients from upstream to predict the microcystins in a lake?
It really depends on your lake. It's each lake responds differently, I think. Other factors would be involved: temperature, depth of the lake, how much light is available in the water column. Is it relatively turbid or is it relatively clear? For example, Lake Erie, it's not we take Maumee River load, so concentration multiplied by flow, and that equal or is proportional to the overall bloom biomass. It doesn't tell us when or where the bloom biomass will be, but it just says there's going to be more microcystins out in the lake. So what you need to do is look into the data you have available for your upstream sources, and then just start looking at correlations between river load and whatever metric of bloom biomass you have. So, but it depends on each lake. If the amount of internal loading, there's internal loading in Lake Erie, but it doesn't change from year to year. Whereas bloom biomass correlates with river load, which correlates with rain. So you also got to count in if your lake has a internal loading or not.
Okay, that makes sense. Thank you. Katie, are the water systems in your study public or private?
They're all public water systems, yeah.
Okay, thank you. There were some other related questions. Where in Ohio were those two?
Yeah, so we're not publishing the public water system information just out of privacy issues, but I will just mention that one of them does pull from Lake Erie, and that we note that in our article, and that is utility B. For those of you that look at the data, you can see that was the source waters that had the most detections of microcystins and also had pretty consistently high biomass.
Okay, thank you. Once your lake has harmful algal blooms, can they eliminate them by reducing nitrogen in the water?
I would say, well, first, how do you reduce nitrogen? I assume what they mean by decreasing nitrogen, not reducing it from nitrate to ammonia. I assume that's what they mean by that. I don't know how you control nitrogen concentrations in the lake. You would have to denitrify it off. But I think the more general question here is, can you remove a bloom from a lake? And the answer is depending on the size of the lake. For Lake Erie, it's so large, you can't go out and scoop up all the microcystis or dump magic chemical X in the lake and kill it all off. You might be able to do that in a golf course pond or a little bit smaller lake. But I think prevention is the best way to stop blooms.
Okay, and do the different microcystin congeners also have different absorption capacity regarding granular activated carbon treatment or removal from the water column?
Can you say the different concentrations? Is that what you said? I'm sorry, can you repeat that?
That's okay. Do the different microcystin congeners also have different absorption capacity regarding granular activated carbon removal from the water column?
Such a good question. We haven't gotten down to that level of congeners. Usually when you're looking at drinking water, you're looking at a total amount. And we don't have, specific effectiveness removal of GAC at, these particular congeners, but a combination, sort of the way we look at it from the drinking water standpoint is, risk reduction. So granular activated carbon is really, certain types and concentrations, and usually, and also other treatment variables like pH and temperature really matter here, especially when you're also looking at the chlorine dosing too. So we tend to talk about it across the board as total microcystins, and it will have those treatment effectiveness will have co-benefits for all congeners.
Thank you. I think this one is for Justin. Did you look into microcystin forecast models that incorporate nitrate concentrations?
We are having, we're working on multiple types of models. One model is a process-based model that Limnotech runs called the Western Lake Erie Ecosystem Model. And that does, that model can forecast nitrate concentrations based on river load. And then those cyanobacteria in the model do respond to nitrate concentration. So yes, that is that's in the work.
Okay. Are any of your grab and go samples associated with an existing solar data buoy?
I don't know if I quite understand that, but there's like 20 different buoys, or maybe there's 10 buoys, but several other stations that like water intake plants have a sensor. We have, I published 2 papers where we have a buoy right outside of Stone Lab that we grab water samples next to several times a week. The first paper we published in Environmental Science and Pollution Research in 2018 shows comparisons between what the buoy says and what water samples say. I can provide a link if that's more, if that information is wanted.
Okay. Yeah, that would be great. Katie, can you clarify when in the process chlorine was used? Was it the same for all, or just varied in concentration?
Yeah, the location was different for all. It depended on their treatment trains when they applied the chlorine. And sometimes we didn't have that kind of information on location. We had just the dose, but we do know that in some cases they were still pre-chlorinating. And for those of you who, know about trying to keep your clarifier working, a lot of them were applying chlorine before the clarification process. So, depending on, and it could vary by day when they where they applied the chlorine. We did still have utilities that were pre-chlorinating because they had to for various other treatment objectives, which is not necessarily what one would consider a great practice for cyanobacteria and cyanotoxins removal, is pre-chlorinating that, because it just lyses the cell and it can become more, have more extracellular toxins that are harder to remove. So great question. I would point towards that article is published, and you can look at some of those details as well. We get into some of that discussion of pre-chlorination if folks want to hear that. I didn't get into it in this, get into the weeds that much on this presentation, but there's a lot more on that. It's a great question.
Katie, can you provide a link to your publication in the chat maybe?
Sure.
Thank you. Okay, this is one that I have myself. What is the relationship between congener and genus and or species?
The congener or variant are, you have a general molecule like microcystin, and then the word congener just means, minor differences among that class of molecule. For microcystin is has 7 amino acids that are arranged in a circle. Okay, and then 2 of those amino acids you can swap out, so you can substitute a leucine for an arginine, and then it changes the congener and changes the toxicity. But different, for example, Microcystis and Planktothrix, different genus of cyanobacteria produce the same congeners. So I don't know if the relationship question is both can multiple genera can produce the same congener.
Got it. Thank you. Do either of you have off-the-shelf study procedures we can use to implement a study in our lakes or rivers?
I would say, I guess your general would be, what are you looking for? And then what sort of funds do you have available to sample the parameters you're looking for? How many sample sites, parameters? So you want to make sure that the data you're collecting can answer what question you're addressing. That sounds like a test question on an exam. What parameters you want to measure: phosphorus, nitrogen, turbidity, and then see how your downstream lake responds. So find a data set or collect your own data set downstream.
Yeah, I would just piggyback off what Justin said here, that I think, especially with our work, the research we did was very specific. So I'm happy to discuss with anyone any of the methods we used and any of the assumptions we used in our analyses. And they can certainly be applied other places. So I'm happy to discuss that, but I think it would really matter on some of the details of what people are looking for.
Great. Well, again, thank you both for your time. I think we got to most of the biggest questions. So, and Justin and Katie's contact information is provided on the slide. So if you have any further questions, feel free to reach out to them. And again, feel free to register for our HAB symposium coming up in January, and subscribe to our events and newsletters on the Algal Bloom Action Team website. Again, thanks everybody for joining us, and have a good rest of your day.